All right, we're going to go ahead and get started. So this last session is in some ways going to be a continuation of what we did with the panel, which is more of an open discussion, but the real purpose of this part is to try and come up with more, let me read what Terry actually sent me. Most discussions have been somewhat meandering, which is fine during exploratory and reactive discussions. For this critical, summative discussion, I think we want to bring the group together to get cohesive answers, and we can walk away from the meeting with. So this really is our task. So, because at the end of the day, all the people that are sitting up here that you can see from Mary over here to George over there, we're going to have to figure out what to do with this, okay? So I'm going to be a little bit more directive in terms of focusing conversation, so we can make sure we cover important topics, but also to make sure we don't get down into a wormhole where we're discussing one particular piece. And so, as I was reflecting on what we've heard yesterday and today, and Teji, you want to go ahead and put up that slide? It struck me that in a lot of the discussions, what we were really beginning to hear about were some considerations for prioritization. In other words, what are the components that we might want to include in a proposed program that would help us to prioritize, say, project A over project B, C, D, because there's a near infinity of projects that we could potentially do. And if we indeed want one study to rule them all, how can we in fact prioritize that so that we can optimize success? And so here are some of the things that I heard over the last couple of days. And the purpose of putting this up there is not to end discussion, but to hopefully begin to, if necessary, explore some of these issues of these criteria, which maybe should have more or less weight, but also to make sure that I haven't missed anything that we think is important. So I'll just quickly read these. So complexity of disease, and one of the things that struck me in Peter's talk this morning was the whole idea that, gosh, we've looked for environmental factors for prostate and we just can't find it. And while that is frustrating in some ways, I think from a design perspective, it makes life a lot easier because we're reducing the variable universe to something that might be more manageable. And if in fact the focus is going to be on polygenic risk scores, then maybe what we're not looking for is something that has huge environmental impact. Again, that's open for discussion. What's the amount of existing data? It costs money to generate data. If we can utilize existing data, we may be able to do some things more efficient, and I heard that point made over and over and over again. In the last talk, we talked a lot about the importance of effect size, and there were several different aspects of this. One is, I think we were all in agreement that if you have a large effect size, that's going to reduce the population that you're going to need to be able to do a study that's adequately powered. So that's an important consideration. We talked about the fact that there are models that are available that can perhaps be used to estimate effect size of different types of polygenic risk scores. We talked about a preventive versus a therapeutic intervention in terms of how that might be able to be measured. We heard that very interesting idea of that we know medications that have a large differential response in different populations. Would that be something where we would see a large effect size that allow a very nice study that would help us to be able to understand why that is happening? The availability, as much as I gave George grief, I did want to recognize that if we do have availability of hard endpoints, that would be something that might prioritize one study over another, David's point. The use of existing longitudinal cohorts. So that's a little different from the existing data issue. So we have cohorts that have been created where we have data that exists over 5, 10, 15, 20 or more years, which allows us to do some in silico pseudo-prospective studies to be able to determine, what is more likely to yield a result if we were to do a prospective study. So one of the aspects, is there available genotype data, or should we generate genotype data? Can we use existing clinical trial data, repurposing it? And then also looking to use the existing longitudinal cohorts to enhance, hopefully, diversity. Multiplex approaches in a population. So if we look at several polygenic risk scores, to Dan's point, and from what Amit had said, maybe we can get to 20 or 30% of a given population that actually would have something that would be of relevance. So is that something that we should be looking at? And then lastly, a point I think that's critically important is the amenability to implementation in real-world healthcare settings. Because again, if all we're doing is focusing on discovery, it'll be one more thing that will just sit on the shelf and won't be used. So if we can find something where it looks like we have infrastructure that would allow us to implement more easily, and again, I'll just use the example of lipid clinics, where we know that we have the ability to do that. And if we could add additional information into the existing infrastructure around a lipid clinic, could that in fact be studied to see what is the impact of adding this information in an already well-characterized setting. Those are the things that I took away. So to start with, any comments, any things that you think are missing, things that are on there that maybe we shouldn't have on there? All right, so for those of you that thought I wasn't paying attention, yes, Nancy.
Well, so one thing that's not on there is sort of the inverse of comments that were brought up earlier around, being much more willing to use the high end of polygenic risk scores to modulate sort of screening frequency than being confident about, say, reducing screening frequency at the low end. And there are some diseases that are appreciated by at least a subset of those working in the disease as being now largely rare variant, large effect, when in fact they also do have a strong polygenic component. And the autism is an example of a space that's really quite fractured in terms of some people believing, that it's mostly rare effect, large variants, others seeing the polygenic, when in fact both are really contributory. It's not clear to me, but I certainly have heard people talk about using only exome sequencing, for example, to make decisions on what variants are contributing to a particular patient, and then the family using that information going forward for reproductive decision making, with no look at the polygenic the liability to autism that may or may not be present. I mean, it's not even considered in a disease where I think we would all recognize that there are both rare variants with large effect and common variants. And just because you have a mutation in one of those genes where rare variants can have a big effect, you're not sure that they do. So I'm wondering if that's a space where there ought not be some further investigations. And I don't know, I mean, I know autism is one example. There may be others in that space.
Yeah, I think we've seen that. I mean, I think it was Peter that presented the slide where you showed the risk curves for different genes associated with hereditary breast cancer.
Yeah.
Where again, BRCA1 and 2 are up in a risk level where you just say, polygenic makes no difference. But if you look at PALB2, ATM, CHEK2, and a whole list of others, you could do that. I think in lipid disorders we have examples, maybe not so much in the loss of function LDLR, but certainly in ApoB, where again we're going to see that same sort of thing, and whether you use your threshold at 2 times the risk or 3 times the risk, where the polygenic will flip you above a determined threshold. I think that's a very interesting application where the likelihood that we would see an effect size would be, I think we would posit would be greater. So I just added polygenic risk altering monogenic disease risk. I'm not editing this in real time, so you won't see what I'm adding here. I also added that we should be looking not only for risk of disease, but also protect in that there's a large protective effect size, that's something we should also potentially consider.
Mark?
Yes.
One other maybe prioritization consideration.
Get a little closer.
It's on, yeah. Would be established evidence of clinical validity and promising hypotheses about clinical utility, because I was kind of struck by Muin's point that he classifies these as tier 3, and I take your point that analytic validity is not an issue with these polygenic risk scores, but I think if we could prioritize scores where the clinical validity data are more established, that would be important.
Yeah, although it was brought to my attention during lunch that the reason that 23andMe is returning just 10 SNPs is that the FDA has said those are the only ones that you actually have the analytic validity data on in terms of the performance of the test. And that is a bit of a sobering comment, given that we know that there have been other NIH studies where FDA has exerted some influence in terms of saying, no, you really can't do that even in the context of research. And if they're going to say, well, you have to validate each of 1 million SNPs on a panel before you can use it, that will bring things to a screeching halt, to say the least. So, but I think, to your point, I will capture both those on this. So established clinical validity and hypothesis for clinical utility. Yes, Naomi.
So the issue of effect size, I'm finding it's getting a bit confused, because I don't think the issue is about the effect size of the polygenic risk score, it's about the effect size of the outcome. So if you've got a longitudinal study of 1,000 people and you take the top 1% for a disease, then you've got 10 people and they've got an increased risk. So going from 1% to 10%, you're still, I'm going to get my sums right. I mean, to measure the outcome in a reasonable time frame, it's very difficult. I have tried to do the sums, and so that's why when we've designed a clinical trial for a grant outcome for polygenic risk score, our outcome has just been simply what's the increase in people going forward for screening. I think that points to what Nancy was saying. Something that's measurable in a big enough sample of people, and actually, when you work through the numbers in a prevention situation, you can start with a very large number to start with and end up with a small number where you're actually measuring your actual effect size.
Yeah. So I've modified that to say effect size to be relevantly defined. So. Right. And that was an interesting point, because I think, as we discussed earlier, I'm trying to keep track of my notes here. One of the important points that was brought up was the question, what question are we trying to answer? And in some ways, I think that there's a bit of a tautology here, because I think the prioritization criteria are going to some degree, inform which disorders would be under consideration, and also to some degree will inform the questions that will need to be answered. But then there's an independent effect, which is to say that there are certain research questions that we consider to be very high priority to be defined, and how does that then influence our prioritization? So those are things I think that will need to be incorporated in our discussions.
So I think also another one is how we characterize the score. So I think positive and negative predictive value is really more important than sensitivity and specificity, which is typically how you characterize your score when you're at least in the discovery phase. Because the positive predictive value is really what's going to determine the effectiveness of the score. And then the other thing I'd say is I think there's a fundamental distinction between prevention and response. So risk scores for preventing disease or modifying behavior to prevent disease versus risk scores that are about response to therapy or response to recognition. I think that's a fundamental.
Okay. Any other comments? Yes, Howard.
I think we're going to see a natural increase in the sophistication of these types of studies. I mean, I still remember when the paper came out that looked at a GWAS for diabetes, and it had over 10,000 SNPs on the chip. It was crazy, the numbers. And then, of course, that goes up to you have whole genome, and then now people are asking questions about what are the markers for diabetes and losing a limb, or and going blind, or and going. And so, predicting higher risk of Crohn's is one thing. Predicting high risk of Crohn's that needs a colectomy is another thing. So we'll see that. I think we need to help expedite the push away from yes/no type of endpoints to a more sophisticated, either continuous variable or at least ordinal, where we start getting severity as part of that. Because really predicting who needs to go to that extreme event. And that's something that can be done with existing cohorts, but having to dig deeper on the phenotyping, as opposed to going in and necessarily having to be prospective about it.
Yes, Eric.
NHGRI has always taken an active stance on genomic studies involving multiple ethnic groups, and it seems like this list needs to have a discussion or consideration of, I don't know what the question exactly is, but do we need to have a risk score for each ancestral group? Are there risk scores that are pan-ancestral groups? I could go on and on, but you get the point. I'm not sure how to word that, but it seems to me that this meeting should, we should note that this, in application of risk scores in quote, the real world setting has many different flavors.
Great.
Including miscellaneous. So I think we need to be heading towards markers that work across, because if we end up with something, if my group never gets in there, if we do only people with Scottish ancestry from the lowlands, not the highlands, and certainly not the Outer Hebrides, then I'm screwed. So we need to do my group. We need to do the Isle of Lewis, not Harris, so that we know what my risk is. I mean, you can't stop. So having markers that are this continuum, because frankly, we're getting more and more of a continuum.
So in some ways, though, I see this as a conundrum, because there's a call to study diverse populations, and we will identify risks that are more population specific. But by the same token, if you think about it from an implementation perspective, if for no other reason than just the pragmatics of saying, what information does a physician have at in front of them that would allow them to say, oh, I should be applying this rather than that. I mean, it becomes intractable. So from an implementation perspective, pan-ethnicity would certainly be more desirable in terms of an implementation research strategy, but that may not be the same answer for discovery strategy.
No, I agree. And I've heard of one guy whose last name was Williams, who's actually Greek.
Yep.
It happens. And so we don't really know what we are to some extent. And so I think getting to the point where to me, it's not an equity issue. There is an equity issue, but to me, it's more a practical, it's not political correctness. We're trying to make it so that you can have objective data about a person and help manage them. And that needs data that it doesn't rely on me deciding that I'm Scottish when actually I have some Irish, don't tell anyone, in me as well.
Yeah.
So,
real diverse.
Sorry, but not the data already is going to tell you that, right? Because one thing is to have the data that you need to create the score, but when you implement it, you will get genetic data that will potentially help you in fine-tune it or modify for that. So you don't need to rely on what you see. It's part of the methodology, I think.
Right. I think the logistical issues are easily surmountable. That a lot of times, if we think, well, there's going to be a separate set of weights for African Americans, and then you could stratify that further by degree of European ancestry or local ancestry. Those are things that I think would be easy to implement in whatever, the clinician is going to see a number. This person's risk in the next 5 years is blah, right? So, but behind that is a complicated algorithm that's sort of, matching to this person and their ancestry as far as I can tell from their genetics. This is my prediction. So that the logistics I'm less worried about. I'm much more worried about the equity. So if it's true that in fact all these, like the true causal variant, the one that's involved in disease mechanism, has more or less the same effect across different ethnicities, and if we had a model that had all those pinned down, Cecile, if you're listening, pardon me. If you had a model that had those all pinned down, then that would be the model we could use for everybody. But we're not going to get there anytime soon. Teasing out what the causal variant is to put in that model is incredibly difficult. And if you're only doing that in, say, European ancestry samples, you would need a huge, huge, huge sample. You're going to get way more bang for your buck, both in terms of building population-specific risk models from having more diverse training sets. And then in turn, I'm totally stealing Alicia's thunder here. Then in turn, you can use that to better refine what the causal variant is. So that seems to be the sort of win-win strategy.
Yeah. I think Cecile's at least appreciates the fact that you were paying attention when she was talking. So.
Can I add to that a little bit?
Yes.
Yeah, so just to, I totally agree with everything Pete said. And then one other point that I want to add is that there's a disproportionate value that comes from studying these minority populations. So for example, in some studies that we've done, we see that having, say, take schizophrenia, for example, where we've looked at East Asian ancestry individuals and have grown up the cohorts there. They're still growing. They're still not the same size as the European ancestry cohorts, but with a third the sample size, we're doing a better job in East Asian individuals than we are with a threefold larger European ancestry cohort. So there's going to be disproportionate benefit, I think, from diversifying our study cohorts, both in terms of pinning down the causal variants and in terms of prediction. We're just going to be able to do a more equitable and more accurate job for everybody with disproportionate benefits as we start to diversify our cohorts, particularly for minority populations, but also for here in the US, European ancestry populations.
Nancy.
But I have to then add, agree with everything they both said, but I have to add, and particularly to add more information on populations of recent African ancestry, where we are very likely to discover additional causal variants. It's the issue is, no matter how well we do in discovery in other populations, there's so much more variation in populations of recent African ancestry that there must also be more functional variation, more and additional factors that affect risk. For anyone who has those variants, they're easier to discover in Africa, but they'll be sprinkled all over the world at lower frequency. And so we arguably gain disproportionately by having more information from populations of recent African ancestry. All populations we want, but we gain disproportionately and will for some time in those populations.
Great. So clearly one of the major gnarly issues that we'll be discussing is this whole, there's all the nuances of this issue, which I think we've captured reasonably well. I wanted to, there are several other questions that were listed in the agenda. I'm not going to read them all, but I think that in terms of getting the fullness of the discussion that we want, there are a couple of other questions that we want to address. And now, one of the aspects that the group should be aware of, and this speaks to several of the points that were made, is that the focus of this will be more in the T3 to T4 range. So looking at things that where we can actually test implementation to try and measure impact on health. And so that then raises some issues. So it's represented, of course, as one of the prioritization considerations, but it also then raises some additional questions, which is, are there thresholds of disease risk or actionability that we should be looking at in terms of implementation? That would be worth spending some time on if people have thoughts about that. I mean, the answer is yes. But I think we want a little bit more depth to that yes, and this is the reason why. Yeah, Bob.
Just thinking about and sort of posing the question: Are we at a point where we're ready to ask those clinical utility questions in a clear way, and or are we ready to just do a nice observational study that will inform future clinical utility studies? Or will we split the difference and say, let's do an observational study and really look very closely at some outcomes that we think are very promising, and sort of lay the groundwork for doing more outcome studies later. I think it's a really interesting landscape.
Iftikhar.
So I agree with, I think, the various choices Bob mentioned, but I think if you take CHD, I think it lends itself very well to this concept because you really necessarily don't need any thresholds. All you need is to incorporate this into existing clinical calculators, and you basically reclassify or re-estimate the risk, and based on that re-estimated refined risk, you take action. So we have very nice algorithms for example, the guidelines for statin treatment and binning of patients in different categories of risk, low, intermediate, high. And generally we know what to do with the low ones and the high ones, but in the intermediate risk category is where we can see a lot of movement back and forth bins, where this the polygenic risk score is going to likely reclassify a substantial proportion of those individuals. So I think it depends on the disease of interest, but I think for CHD, it's very beautifully lends itself to widespread application in the clinic.
Yeah, George.
Mark, I strongly agree with that, and I think that's where I might put Eric on the spot, maybe to comment on what the experience in top med has been or how you're thinking about that, because here we have the unique opportunity of having these cohorts for decades, where several of them we have some very good deep phenotyping also. And so really, even if the only key clinical question we addressed was what do we do with individuals with intermediate risk now, it would be extremely beneficial and very, very actionable.
Timely, too, since we've got a top med meeting starting tomorrow. We're going to be talking about next steps. So, so George has put it on to Eric. George, who's from NHLBI, says we should be doing heart disease, which is shocking to me. But
I really didn't say that. But
no, I know. I was reading between the lines.
I'm going to second the heart disease comment, but not because of the NHLBI connection, but because of the one Iftikhar brought up. And I had one slide, if you recall. I mean, there are standard practice guidelines that put people into buckets according to risk. And as some of the people around this table know, establishing those guidelines is a bloody exercise. And so to think that, some geeky geneticists are going to redefine those buckets, I think is naive. However, to redefine what goes into the risk calculation is imminently doable and is periodically revisited. And so I think, basically many programs, TopMed, CCDG, are discovering genes for these various common disease conditions. They can go into existing risk calculations. That's number one. I'll just parenthetically say it's multi-ethnic too. That's important. The Hebrides is not represented. However, but again, I think the question that can be attacked is the one you just brought up: what to do with people in these intermediate categories, number one. And then number two, as you said, these people are followed longitudinally, so you don't have to use surrogate endpoints of lipid lowering, for example, alone. You can actually see, you'd have to do the power calculation, you can see outcomes, and you can see multiple cardiovascular related outcomes. So I think it's a reasonable, I'm not sure it gets the label low-hanging fruit, but it's certainly something that's imminently doable.
Yeah, and I agree, and I think that was something that was missed on our prioritization consideration, so I've added that to that slide because I think that seems quite logical, because really everything is already defined around it, and so and we were whispering to ourselves earlier about the idea that we're really talking about a scenario of clinical equipoise where in an intermediate risk you can choose to do or not to do. And that's a perfect place to really identify where there's potential benefit at essentially no harm, because people are already choosing to kind of randomize just based on practice patterns.
And one other comment, I'm sorry, and I'll be quiet. I also think it's a place where you can grapple with this implementation science question, which I think in some ways is the hardest question. I think the genetics is going to come along. but how you actually get it into the clinic and the steps and the workflow and insurance, all that's going to be critically important. And I think cardiovascular, and I guess I'm biased, lipids and hypertension alone is a good place to answer some of those implementation science questions.
Okay, George.
Mark, I was also going to add before we forget, in your last bullet, it's pretty loaded. There are a lot of important issues we talked about here. It may not be a bad idea to have a couple of sub-bullets. I think the diverse ancestry is important, but just from the last comment, issues around trust in specific populations that would lead to implementation in real-world health settings would be key.
Okay, great, thank you. All right, another question that came up was, and I think we've just touched on a number of those things, but if we wanted to explore this further, in what contexts should genomic risk prediction be implemented and how? So I think we had a really nice discussion about the use of places where we're already using risk calculators to stratify populations, that a lot of these are in use in cardiovascular settings. Are there other contexts that we could consider to explore as we begin to think about how to design a study? Dan?
Well, there are lots of other contexts, and I'm not an expert in any of them. But so when you say cardiovascular, you mean atherosclerosis. So I'll just make sure that we add atrial fibrillation to that list. Patrick has left already, so I'll be his surrogate for a while. There's promise for polygenic risk scores in some guise to help guide therapy or identify high-risk people in virtually every disease domain, in neuropsychiatric disease, in inflammatory diseases of various kinds, in predicting certain adverse drug events. There's an emerging story around that, around various adverse drug events. So I think that it would take.
You keep touching this.
It would take a.
And don't touch me either.
It never occurred to me. It would take a longer meeting than this to sort of make a catalog, but I think it probably would be worthwhile to sort of make sure that before we make big decisions, that to survey the literature. I've heard stories about osteoporosis. I've heard stories about gynecologic disease. So I think that there's a lot of opportunity. The reason atherosclerosis is so appealing is because we know that it's a common disease that's polygenic, and it's been extensively studied. So the foundational GWAS is available, but the foundational GWAS for many other things are coming online or available as well. So I think that thinking about the places where therapies are expensive, therapies may be high risk, maybe one way to think about that.
Diabetes.
Okay.
So I do agree with what Carlos said yesterday, that if you find people with low PRS for diabetes and the intervention is stopping their metformin, I think that's the sort of long run for a short slide, or long slide for a short run, or whatever the right phrase is. That's probably not where I would spend a lot of effort.
Well, and there was also the, I think it was one of the presentations this morning where it showed, here's all the different ways that we can classify risk, and you look at the, clinical factors that we can easily get at, which had like the third highest area under the curve, and the delta that you got from adding polygenic to that. I mean, that's not a disease I would think would be very good to study, because you're really not going to be able to detect very much. It's the ones where we're really not getting much out of our current clinical factors, but we're still trying to do some sort of a population stratification where this could be the biggest, could have a bigger impact. Nancy.
So I know a point was made this morning that neuropsychiatric disease was not a good place to start. And use schizophrenia as an example, but for all the reasons that the prostate cancer was considered a good model. That is, there's no there's very little else that is predictive of the risk of prostate cancer. The same, if not more, is true for neuropsychiatric disease. We have few good predictors, we have no biomarkers, and both the burden to healthcare for at least some of the neuropsychiatric diagnoses and the length of diagnostic odyssey. For bipolar disorder, the diagnostic odyssey is measured in years, and 5 to 7 years is not uncommon for a period of time where people go from specialist to specialist seeking improvement for their pain and conditions. And the opportunity to diagnose those patients more rapidly and improve their lives, reduce the burden on the healthcare system, and reduce the number of suicides that would otherwise occur seems like a win in those contexts. So I wouldn't dismiss the utility of polygenic risk scores in conjunction with other kinds of prediction models, because you gain, there's already data from using patterns of healthcare usage in prediction of a neuropsychiatric diagnosis over the near term.
So the utility that you're looking at is a diagnostic utility as opposed to a interventional...
A diagnostic odyssey, sort of.
Yeah, reducing, which of course that's an interesting, I think we all recognize, at least those of us in genetics recognize that this is really an important thing. Frankly, it's extremely hard to value. And so if we think about the uptake on the, it's been a less than compelling argument for our friends on the payer side to say, we can, reduce your diagnostic odyssey, even though we can quantify pretty well how much money they've been spending on tests that are relatively ineffective. But be that as it may, I think that these are all good points that we should consider, that it may not be about an interventional thing, but it may be about actually achieving diagnosis, which is still the beginning point for all we want to do from interventions. So. Let me go back to the list of questions. I think we've covered most of them in the various discussions, but there's one that I want to tee up as sort of maybe the last discussion point, and that is what would be considered the critical knowledge gaps? What are the known unknowns? We know there are going to be unknown unknowns, but what are the known unknowns? And are there ones in particular that haven't been explicated in all of the presentations that have gone before? Yeah.
So one interesting one has to do with the age of the cohorts in which predictive models are built. And actually it's of particular interest perhaps in cardiovascular disease, where some of the cohorts are older and many of their risks may have been ameliorated by statin therapies and other anti-atherosclerotic therapies. It's a really interesting question of whether the risks measured and estimated in a much older cohort or older cohorts, and some of the genetic data come from really old collections, dating back even into the linkage days, you must remember, how relevant those continue to be for new event prediction, for example. And
okay, so I was missing, because when you were talking about old, I was thinking about the age of the cohort, but it's not the age of the people in the cohort, it's actually the age of when the data were actually originally collected.
The cohort of the cohort.
Yeah, the cohort of the cohort, yeah.
What is the gap, though? What's the gap that you're referring to, that we're not using
I would
those?
characterize it as how applicable are risk prediction models that were created using data from the 60s and 70s to the 2000s and 2010s. Is that a fair way of?
Well, you know, even in a biobank or data sets that go back 20 years, when you're looking at those as your case set and building predictions around those, things have changed. And you know, a great example is the lowered risk for ApoE carriage in Alzheimer's in successive generations of the Framingham cohort that benefited from a whole variety of changes in healthcare because of Framingham. But it's affecting the estimated effect size for being a carrier of ApoE and the development of Alzheimer's.
Okay, great. Other knowledge gaps that we really haven't discussed or articulated very well? Yes.
George raised this, other people have raised this, but I think the implementation is a known unknown. Like, how does this actually get into the clinic?
Yes.
I think sort of along those lines too, the receptiveness of providers in practice to using something that's genetic in basis as opposed to something that they've been taught in their training, you know, clinical scores.
Yeah.
So,
although, to be fair, a number of the currently funded studies like Ignite and Emerge and some individual institutional studies are really looking at that sort of provider acceptance, and in some cases it's being extended to providing genetic information that they didn't in fact order in the first place. So I think some of that will hopefully within the time frame of planning for the study we'll have more knowledge than we currently have
so
about that.
The outcome is trying to understand how to reduce that barrier.
Right, exactly. Eric.
Obviously, in planning this meeting, you put a lot of thought into other omics, and we spent a lot of time this morning, but in this kind of wind-up, we haven't talked a lot about the role of other omics. And I don't really know where the reach of NHGRI stops, but it does seem like it's very possible that these other omics are going to be more predictive risk scores than genetic risk scores. And as much as you can, again, that's for people, you know, in the middle there to decide if it's an NHGRI measurement, but it does seem like we should consider the role of other omics in prediction.
Well, we did discuss that last night at our sort of midpoint dinner, and I think Terry would say that, you know, the extent would go downstream from genome, that if there are you know, whether it's a transcriptome or a proteome or something that, you know, is related. Stop.
So no, we sort of define our domain as the nucleic acid. The gentleman to my left may correct me, but really, you know, so RNA, transcriptome, cool, epigenome, great, proteome, it's getting a little bit far from us, even though proteins are intimately involved, as you made the point, in transcription and other things. But we're kind of, that's where we get a little bit further from us. Would you agree, Doctor?
Yes.
Yes.
So good. Terry is relieved that he agreed, although they've had that conversation any number of times. My reflection on the session this morning, which was fascinating, was that we have much more to learn in the discovery space there, and if this is more you know, focused on, you know, what might be ready in the near term from our implementation, that, you know, that might not be a primary focus, although I think it would be potentially interesting to say, coupled with, you know, what we're using from polygenic risk scores, are there other things that we should be measuring that might be more proximate to the outcomes of interest? So.
Yeah,
yes.
No, I mean, come on. So Funmi's example of going to yearly mammograms to begin to catch the tumors, and frankly, I've seen people now talking about doing or imaging every 6 months because of the very high risk and the fact these tumors develop so rapidly. If that's not an area where people ought to be looking at serial transcriptomes which don't cause any harm in comparison with, you know, imaging every 6 months, I don't know what is. I would argue that in any context where we're doing this very careful monitoring that has the potential to add any harm to patients over the long run, we ought to be considering serial transcript checking whether serial transcriptomics could do a better job of identifying the early stages of those conditions. And I mean, transcriptomics, the potential that the epigenomics would, I think those are great examples of places where we ought to look with some of the other easily deployed omics to see whether those are as good or better than what we're doing now that have harms associated with them. Blood biomarkers, yeah.
So within the context of an implementation where there's an intervention, then looking at what are the ancillary things that could be measured beyond what we currently understand could be a place where we could look at some of the extended, and that seems reasonable. Although again, the scope would be something that would have to be addressed, because it's not that it's not an important question. In fact, we're doing research on that at Geisinger now. But trying to get everything under one umbrella can be difficult. But I think that's a really good point about if we're going to go beyond the genome, how we deploy that should be in a place where it's likely to provide very useful information in a relatively short time. Yeah.
Is there a way when we develop the polygenic risk score, or when we implement that risk score, that we, and that's probably something for the statisticians more than me, but that we use that information on omics, for example, when we develop weights? Can we weight these SNP differently if they're EQTL or MQTL, or they have some biological relevance? Would that seem to be a way of incorporating some of that omic information?
So let me try and rephrase that and make sure I captured what I think I heard you say. In the context of a knowledge gap, we've talked about different ways that you can combine genetic risk scores with other features, but we're doing it sort of empirically without a lot of evidence, and that we've not really actually studied what's the best way to combine this information to really get the maximum effort. So it's really about the methodology of assessing the combinatorial aspects of risk scores. Is that fair?
Yes, that's fair. So, and then incorporating that knowledge of omics into that.
Yeah. Okay. Yes, Marc.
So coming from a healthcare system where we reached to 100,000 patients to take up our genetic tests, which will lead to a genetic risk score calculation, 93,000 patients declined to participate. We only have 7,000. And there is a concern in the patient population about potential insurance risks and other things. Of course, this could be a research topic to see if this actually does make a big difference, but it's also a policy topic for NHGRI and NIH to make sure that the patients do not have these concerns going forward. GINA is a great law that helps the patients make this decision, but there's life insurance, disability insurance, and other insurances that the patients feel very concerned about.
Great, thank you. Naomi.
That's just a terrible percentage. So what was the question they were actually asked?
We have a test called Sanford CHIP that has an 8-gene panel of pharmacogenetic testing and SNP CHIP, essentially the global screening array that the 23andMe uses to basically take this test as a clinical test and return results for the ACMG59 genes. And over 100,000 patients were asked to participate. 7,000 so far has accepted in that cohort. 93,000, there's a good portion of that the patients express concerns about life insurance, disability insurance, and other long-term financial complications.
So I'm going to extract from that one piece that I would consider to be a known unknown. And And it's nice, Murat, that you guys are actually collecting reasons for decline. But in most studies, when people decline to participate, that's the end of it, and we don't get additional information about why they're choosing not to. And I think you're right that there may be some very important signal about, you know, what would constitute it. And so while it wouldn't be major focus, say, of the project, one could envision designing it in such a way that when consent was obtained, if for those that decline, what are the driving factors related to that? So I think we could characterize that as something that at the present time is unknown. But I would also echo, I think, what Naomi is saying, which is your experience is clearly very different from many others, including ours. So.
It made me think this discussion is that, you know, if you are asking people, do you want your test results back, what actually should we be asking people? Because maybe we shouldn't be saying, we'll give you your risk score back, which requires interpretation. Should we just say, would you like to know if you're in the top 1% because that's actionable? And I think that would be an interesting question to see if people would change, you know, have a different decision process on that, because then, you know, insurance, you know, if you actually know you've got a very, very high risk, then you might take, you know, your considerations about insurance might be different than if it's just knowing that you've got a test on file.
So to clarify a few things, we're not returning polygenic risk scores. What we're returning is the 8-gene PGX and the ACMG59 positive findings. And the second thing is that we haven't asked all 100,000 patients why they didn't take the test, but there is a portion, I don't know the exact number, but there's a portion that start the consenting process and leave in the middle or have a question about their consent. Then our genetic counselors reach out to those patients and have a discussion with them, and that's the feedback we get from that cohort.
So I think we've got a pretty good sense there, and actually it raised an idea in terms of the prioritization that we talked about yesterday. I think it might have been George's talk about the idea that we need more patient-centered measures related to this. I know you put that up on your slide today as well, and that's something that we should also be considering in terms of an area of prioritization. So, and clearly that would fit within there. We're getting close to the end here. Eric.
In terms of known unknowns, maybe it was picked up. It's on.
Into the microphone, yeah.
Yes, I can't get much closer.
But you have to talk into the microphone.
I heard a couple threads on this, but I wanted to just point out that it seems to me that one of the differences we face in this circumstance is that the underlying data will change. Well, we'll get a score now, but inevitably that score is going to change. The underlying data, there's going to be knowledge that's going to accumulate, and if we're going to have issues with healthcare professionals accepting a score, they're going to also have to accept the fact that that score is going to, the ground's going to shift over time with this modality. And that's going to be a slightly different circumstance than the introduction of a well-refined clinical test that stays stable over time. And we want it to shift, we want it to improve because the knowledge will improve and so forth. So I would just mark that as another challenge ahead of us.
Great. Okay, so Terry is about to get set up to finish things up. So it'll take her about 5 minutes, I think.
No, I don't think so.
You don't think? It'll take her about 3 minutes. So you have 2 minutes.
4 seconds.
If there's any last-minute dying questions before Terry does her inestimable sum-up of the last 2 days, we'll take them now. Now, the fact that you didn't get a chance necessarily to say something now, or if something occurs to you in the future, please contact TAG probably, or meeting organizers, and just send the information in, and we can do it. So the last word before Terry.
Yeah, just one point about the data to collect, the collection of new data, especially new populations that are not European, to address this issue of diversity. You know, some of the microarrays that we're using today were designed based on European, very small amounts of SNP data from Europeans, and that has the impact of bias the collection of the data. So we need to think about this when we're designing these new diversity studies. If we do sequencing, there's no problem. But if we use microarrays, I think it's important to know what's the impact of that old design on collecting data outside of European populations.
Great.
And the other thing is there has been some interest in using low coverage sequencing to generate scores, because that presumably gets us to the same cost of a microarray without this bias. But I wonder if there's no bias by the imputation that is used to recover those genotypes back. So I think that's another area that, although there is some entities that are very enthusiastic about that, I don't know what's the impact in terms of biases of populations and transportability to populations.
So that's sort of a technical issue, and I guess one of the questions that would occur to me with that is, one of the issues that has prevented us from doing large-scale sequencing studies is just the cost. And so the question would be is what's the differential between, running an array versus even a low coverage genome? I suspect it's still probably a couple of orders of magnitude, which has a necessary impact on population size for studies.
Do you mind if I jump in on that? So we've done some analyses of the costs and the differences in different arrays, and the cost all in with all labor and all sorts of factors that consider overhead, how much it costs to keep the lights on for something like the global screening array, which is a misnomer, it's not a global array, is about, say, $65 to $70 a sample. And for low coverage sequencing from a company like GenCove, for example, that does something like 1X, it's $90 a sample. There's differences, but they're not huge.
Not as big as they used to be. Okay, so that's an interesting point to put on the table at least. So.
Yeah. We've talked a lot about ancestry. I'm doing everything I can to stop this. We've talked a lot about ancestry, and there's the European ancestry, and Howard alluded to the idea that he thinks it needs to be island specific. And then there's African ancestry subjects, but then there's the question of what about African Americans who are admixed to a variable degree, and do you construct a genetic risk or a genetic risk score based on an average African American, whatever the hell that is, or do you go and look at each LD block and decide whether they're European or African at that particular site and then choose the right SNP to stick into the score, or what? So I'm looking at Eric and Naomi and Nancy. How do we do that?
Well, ultimately, I think what you said is what will happen is that there'll be, you'll decide which ancestry is the block from, and then you'll get a risk score for ancestry-based risk score. But
carefully.
That's all under the hood. I mean,
yeah.
You don't calculate them now. What do you care?
As long as it works. I mean,
that's right. It is a much harder calculation for a whole bunch of reasons. In admixed populations, in populations with much less linkage to disequilibrium, it's a much more complicated calculation. But in the end, and it'll get better and better, and but it'll all be under the hood for all of it.
Yeah, just adding to that, the structure is like a really, really challenging issue to overcome. The structure is not just the mosaic of the blocks locally within a chromosome, but also cross-chromosome. That creates some really big challenges for stratification where sort of environment and genes kind of like intersect. There's some methods that currently exist in that space. Multipred from Alcus Price's group is one, but it's a really active area of development. No one's yet using the local mosaic of ancestry to build predictors, at least in the human genetic space, but Naomi probably knows better from some of the agricultural literature. But yeah, we should definitely be building methods there.
That was part of the point I was trying to make, though, is that we're never going to be able to get all the populations we want. So rather than designing a study for African Americans, and then, maybe we should do a study for Hispanics, whatever that is. Let's try to see if we can prove these things to be useful, or do discovery from admixed populations, but design it properly. So it's not like 10% of our clinical trials at the NCI are African Americans. Well, we'll never say anything definitive about African Americans from those studies. So we need to power it to answer the question. But right now, it's all convenience data sets, and we're stuck with what we have.
All right, so it looks like we're going to try.
No, but I think the problem with the previous discussion is that the genetics part of that is actually, I don't think, the most difficult. It's like Nancy said, the science is coming along, the calculation will occur, but don't forget, those risk scores have to be done in absolute risk. You have to go from relative risk to absolute risk. And that's where I think Howard's contention, I'll use that word, has the most weight. You're never going to be able to develop an absolute risk equation for every pocket of humans around the world. We just don't have the data. So how we develop those absolute risk calculations, even the way we do it now is silly. We think all African Americans are the same or all European Americans are the same. It's just ridiculous. But nonetheless, it's the best we can do at this point in time. So I don't know how to do it, but I think some kind of a pan ancestry risk score probably is necessary, but I'm not sure it's feasible. Yeah.
But it does add to our known unknowns in the sense that, one other area, as you just referenced, is the idea that we do need to convert these to absolute risk scores, and we're not necessarily confident in terms of how exactly to do that, irrespective of the population issue that was brought up. So, blessedly, Terry is now online, and so we'll turn it over to you, Terry.